Releasing this video where we talk about more DNA replication and we just tackle DNA code of life as a whole. Now, boys and girls, here we'll be looking at a question that asks us, basically tells us that this is a DNA profile of 6 family members, and essentially the parents Zinhle and Ayanda have 4 children, and 2 of them are biological and 2 of them are actually adopted. And they're asking us in question 2.2.1 as we get started. I'm getting straight to the point now. You see that my videos are less hello and buys, and yeah, we just need to get straight to the point at this point in time. So essentially it says to us, which 2 children are the biological offspring to Zinhle and Ayanda? Now I want us to understand something, boys and girls. When a person, or when you are born and you are made, or you are, created, you will have 50% of your DNA from your mom and 50% of your DNA from your dad. That's just how it works. So 50% is dad, 50% is mom. If there's a bit of discrepancy there, you must know that, okay, likely not my father. Okay. Now I want us to quickly look at this question and see what I mean when applying the knowledge that I just gave to you now. So let's see. The patterns of the parents must match that of the children. Okay? Let's look at who matches who. Let's first start with Zinhle, which is the mother. I'm going to assume. Okay, so let's start with the first pattern. The first bar, we see that, Lindiwe has one that matches. So let's just make a tick here. The other ones don't match. Let's look at Zinhle's second bar. Zinhle's second bar, we can see that Bandile and Lindiwe seem to be matching. Okay, it's fine. Let's go to the third bar of Zinhle and see what matches. It has to be moving in a line. Let's see. Bandile matches. Okay, Bandile matches. Perfect. And Bandile is the only one that matches this time. And then let's go to the fourth one. And the fourth one, we see that, wait a minute. Bandile matches. Okay. And let's go to the fifth one. In the fifth one, we can see, Lindiwe matches. And Lindiwe is the only one. And then in the last one, we could see that, okay, Lindiwe matches and Bandile match. Okay. Perfect. That being said, I want us to then look at the father, who is Ayanda. Ayanda, the first bar of Ayanda matches that of Lindiwe, and that's it. It has to be identical, exactly identical. The second bar of Ayanda, let's put a tick here, matches that of Bandile. Okay, perfect. The third bar of Ayanda matches that of Lindiwe and that of Bandile. Okay, they're in the same line. You would use in this case, you would use your ruler, boys and girls. And then from there on, let me just quickly see if I can't show you guys how I'd actually. Let me see if I can't get a ruler here quickly. 1 second. Yeah. Okay. Might seem a bit embarrassing, but it's okay. It's fine. That is fine. You'd use a ruler and just go accordingly to make sure that you're correct, to increase the size, to increase the accuracy of what you're saying. Let's move on to the fourth bar. The fourth bar of Ayanda matches that of Lindiwe, not that of Gugu, even though they're in the same line, but that of Bandile, and not that of Litswe, because you could see the Litswe one moves. Litswe, is a bit up. Okay. Now we said, what did we say? We said that 50% mom and it has to be 50% mom and 50%, your DNA is 50% mom and 50% dad, right? 50% maternal and 50% paternal. Okay. So it says, which 2 children are the biological offspring of Zinhle and Ayanda? Ayanda has how many? Let's look at Lindiwe first. Lindiwe has 1, 2, 3, 4, 5, 6 bars. So it means 3 must be father, 3 must be mother. And we could see that, okay, 3 is father and 3 is mother. Bandile has 1, 2, 3, 4, 5, 6. 6 bars. 3 mother, 3 father. Makes sense. The ratio does make sense. So therefore, the children are Lindiwe and Bandile. Bandile and Lindiwe. And Lindiwe. 2.2.2, boys and girls, as we said, it says, give an explanation for your answer using evidence from the DNA profiles. You would say 50% of the offspring's bars in the DNA profile match that of, who are the parents? Zinhle, and 50% of the offspring's bars match that of Ayanda. And as we know, you just state there, an offspring inherits 50% of their genes from the maternal side and 50% from the paternal side. So you get a mark for stating that 50% of the offspring's DNA matches Zinhle, and 50% of the mother, and you get a mark again for stating the fact that, as we know, an offspring inherits 50% from the maternal and 50% from the paternal. Okay. And then question 2.2.3, quick and easy. It states, apart from paternity testing, state 2 ways in which DNA profiling can be used. That's when you go back to the notes I gave you. Apart from paternity testing, what are the uses of DNA profiling? And I did give them to you, yeah? Excuse me, boys and girls. Here we are. So it's apart from paternity disputes, we can say to identify dead bodies and matching tissues for organ donating. So let's quickly write it here. You can see exactly, just go to the notes. My answers are in the notes. Learn the notes that I give you. Very simple. So it's to identify dead bodies, and last but not least, to match tissues for organ donations. Right, perfect. I want us to quickly just look at this question. Let's look at the multiple choice questions, and then I'll release a separate video whereby we do question 4.1.1. Now it says, yeah, question 1.1.1. I want you to always double check your answers for multiple choice, because people tend to get happy and that's where you easily make mistakes. It says the shape of the DNA molecule was discovered by who? Is it Rosalind Franklin? What did Rosalind Franklin do? I stated that Rosalind Franklin, if you look at your notes that I've given you again, Rosalind Franklin took the X-ray photo of DNA. And then Watson and Crick discovered the shape of DNA being double helical or double helix. So now the shape, let's quickly look at the question. It says Franklin using the evidence obtained from Watson and Crick. We know that A isn't our option because Watson and Crick stole Franklin's work. And then it says B, Franklin working independently of anyone else. Nope. And then it says C, Watson and Crick working independently of anyone else. No. And we can therefore say that the answer is C. What does C? The answer is D. What does D say? It says Watson and Crick, perfect, using some evidence obtained from Franklin. That's perfect. So we get a D. The answer is D. So you get 2 marks for this one. The next question from our past paper states that messenger mRNA consists of 300 nitrogenous bases, right? The maximum number of amino acids that can be coded for is what? What is the maximum number of amino acids that can be coded for? Let's think about it for a bit. Remember, we will have a nucleotide, right? So we'd have a nitrogenous base. For example, in a nucleotide would have a nitrogenous base, and essentially the nitrogenous base bonds to another nitrogenous base. And in this sense, they're saying that mRNA consists of 300 nitrogenous bases, right? So if mRNA has 300, then tRNA, what would tRNA also have? It would also have 300 nitrogenous bases. And then from there on, that being said, the maximum, it says calculate the max, what would the maximum number of amino acids that it could, what's called again, amino acids that it can code for. What would that be? We know that an amino acid is formed from what? Three nitrogenous bases. So therefore we'd say 300 divided by 3, and that will give us 100. So C is 100. 1.1.6 is 100. Question 1.3.3, it says the code for amino acids on mRNA. The code for amino acids on mRNA. mRNA, we said, is a codon, not an anticodon. So in this sense, A is the answer. Question 1.4.1. 1.4.1, boys and girls, states to us, it says, it says, identify molecules W and U. So W and U, let's go, okay. What is W? We're looking at W. W, you can see there's thymine, there's a phosphate group, and a sugar molecule, W. So that would be a what? That's a nucleotide. What is U? U is DNA. It's DNA molecule that is double helical in shape. Now B asks us to identify the parts on W, or parts of the molecule W, labeled X and Y. So X and Y. What would we have in X? X, when remember our drawing of the nucleotide. X would be your, what would X be, boys and girls? It would be your phosphate group. And Y would be your sugar molecule. And then what type of bond is Z? We said that nitrogenous bases in DNA are linked together by weak hydrogen bonds. That being said, boys and girls, I want us to move to the next part of the question. Question D says, identify nitrogenous base V. Identify nitrogenous base V. That's what question D says. Nitrogenous base V. Where's V? Yes, here we go. Nitrogenous base V. So we see that this would obviously bond to V, and they said that V for us is molecule. I guess just listen to what I'm saying. Apologies. So we see V is bonded to this molecule that I've highlighted in blue. This molecule, essentially. And that being said, it's in DNA, and the nitrogenous base is thymine. And they're asking us now, identify the nitrogenous base V. So what does thymine bond itself to? Thymine bonds itself to what? Adenine, because apples and trees, cars and garages, as if you guys would remember the rhyme that I taught you guys. Now, let's go on to question 1.4.2. 1.4.2, it says, Where in the cell does this process take place? DNA replication takes place in the nucleus. During what? During interphase. Okay, let's see what question 1.4.3 asks us. It says, name the phase of the cell cycle where replication takes place. It takes place what? During interphase. If you were to listen and if you were to remember that I did teach this and I did say this in the beginning of the first lesson. If you can't remember this, go back and learn, go back and learn, go back and learn. Now let's quickly do question 3.